Hello everyone, I am Amit Jaiswal from IIT Mandi. Today I will present a brief overview of our recently supported ANRF-ARG project on chiral gold nanoparticles for sensing, imaging and light-guided therapy. At the outset, I sincerely thank the Anusandhan National Research Foundation, Government of India for supporting this research and IIT Mandi for providing the academic and research environment to pursue this work. Gold nanoparticles are extremely tiny particles, much smaller than a human cell, but they interact very strongly with light. When we introduce a special twist or handedness into them, they become chiral nanoparticles. This presentation explains how such light-responsive materials may help future healthcare technologies. Before moving to the main ANRF project, I would like to briefly introduce the broader research focus of our lab. At AG Lab NanoBio, we work at the interface of nanomaterials and biomaterials, with the aim of designing advanced materials to address biomedical challenges. On the nanomaterial side, we develop plasmonic nanostructures such as anisotropic nanoparticles, core shell nanostructures, and nanorattles. These materials interact strongly with light and are explored for sensing, imaging, stimuli-responsive therapy, and photothermal therapy. On the biomaterial side, we work with cationic polysaccharides and 2D nanosheet to develop materials for antimicrobial applications, wound healing, and hemostasis. So the common theme of our lab is to design functional materials that can either detect disease signals, respond to light, fight infection, or support tissue repair. The current ANRF support work on chiral gold nanoparticles builds directly on this foundation. Gold nanoparticles can be useful in healthcare in three broad ways. First, they can help us sense very small amount of molecules. Many disease-related molecules are present at extremely low levels, especially at the early stages. Gold nanoparticles can amplify weak molecular Raman signals and make them easier to detect. Second, they can help in imaging by improving contrast so that abnormal tissues or cells can be seen more clearly. Third, they can help in therapy. When near-infrared light is shown on them, they can convert light into localized heat, which may be useful for targeted treatment. So the same family of nanomaterials can potentially support detection, visualization, and therapy. This slide introduces the concept of chirality. Chirality simply means handedness. Our left and right hands look similar, but they are not exactly the same. In the same way, nanoscale materials can also have left-handed and right-handed structure. Why this is important? Because chiral nanoparticles can interact differently with different types of circularly polarized light. Some light waves rotate in one direction while the others rotate in the opposite direction. This difference can be measured using an optical technique called circular dichroism. In simple words, chirality gives nanoparticles a unique optical identity which can be useful for advanced sensing and biomedical applications. Most of the molecules has a unique Raman signal, almost like a fingerprint. However, when the molecule is present in a very small amount, this fingerprint is often too weak to be detected. Chiral gold nanoparticles can act like tiny optical antennas. When molecules come close to them, weak molecular signals can become much stronger. This can help detect faint chemical or biological signatures that may otherwise remain hidden. Such sensing approaches may be useful for detecting disease biomarkers, toxins, contaminants, or other trace-level molecules. The key message is that these nanoparticles can help us see molecular signals that ordinary optical tools may miss. This slide shows that the shape of gold nanoparticle is very important. A gold sphere, rod, cube, star, or chiral particle does not absorb light in exactly the same way. Each shape has its own optical response. This property is known as localized surface plasmon resonance, or LSPR. In simple terms, when light falls on a gold nanoparticle, the electron on its surface move or oscillate together. The wavelength at which this movement is strongest depends on the particle's shape and size. By tuning the shape, we can tune the optical response of these particles for different applications. Near-infrared light or NIR light is especially important in biomedicine. Visible light cannot travel very deep into tissues because it gets scattered and absorbed near the surface. NIR light can penetrate deeper into biological tissues. This makes NIR light useful for bioimaging because it can help collect information from deeper regions. It is also useful for photothermal therapy, where nanoparticles absorb NIR light and convert it into heat. Another advantage is that NIR light generally gives lower background interference, which can improve imaging clarity. Therefore, combining NIR light with gold nanoparticles provides a promising route for deeper imaging and more localized treatment. This slide explains photoacoustic imaging in simple language. First, near-infrared light enters the tissue and reaches the target region where nanoparticles are present. Second, the nanoparticles absorb this light and generate tiny heat changes. These heat changes produce ultrasound waves. Third, those ultrasound waves are detected and converted into an image. So photoacoustic imaging works by sending in light and receiving sound. This combination is powerful because light provides contrast while ultrasound helps with deeper tissue imaging. When nanoparticles are placed in biological environment, they face several challenges. They may aggregate, interact with proteins, or be cleared rapidly by the body. One way to address this is biomimicry, which means learning from the nature. Here, nanoparticles can be coated with a cell membrane-inspired layer. This coating may help them remain stable, reduce unwanted biological interactions, and improve compatibility. In simple terms, we try to make engineered nanoparticles behave more like natural biological objects, so they can function better in a complex biological system. This slide shows the basic idea of photothermal therapy. First, nanoparticles are delivered to the target region. Then NIR light is applied externally. The light reaches the nanoparticle and is absorbed by them. The nanoparticle then converts this absorbed light into heat. Ideally, this heat is produced mainly at the target site where the particles are present. The aim of this approach is localized treatment, heating the diseased area while minimizing damage to the surrounding healthy tissues. This is one reason why light-responsive nanoparticles are being explored globally for future minimally invasive treatment strategies. This work builds on our group's foundation in three connected areas: materials, optics, and biology. From the material side, we design plasmonic nanostructures. From the optics side, we study how they interact with light. From the biology side, we explore their use in sensing, imaging, and therapy. Our previous work in plasmonic nanomaterials, surge sensing, nanobio interfaces, and photothermal therapy applications provides the foundation for this ANRF-supported research direction. To conclude, this work aims to translate light-responsive chiral gold nanoparticles into future tools for healthcare. The broader vision is to develop materials that can help in earlier detection of disease signals, clearer imaging, and more localized light-guided therapy. This project also contributes to strengthening indigenous capability in advanced nanomedicine and biomedical nanotechnology. I once again sincerely acknowledge the Anusandhan National Research Foundation, Government of India, for supporting this research through the ARG scheme, and thank IIT Mandi for the institutional support. Jai Hind.